Hello students, let's discuss about enzymes involved in prokaryotic replication. Introduction. DNA replication is a process that allows production of exact same copy of chromosomes to be passed on to the next generation. The process of DNA replication in prokaryotes requires participation of a number of enzymes such as DNA polymerases, Helices, Topoisomerase, single-strand binding proteins, Primase and Ligase. The well-controlled choreography of the enzymes makes the process of DNA replication possible. Now let us go into detail about the enzymes in prokaryotic replication. Let's discuss about DNA polymerase. DNA polymerase is the main enzyme that is involved in DNA replication. Arthur Kornberg discovered the first DNA polymerase and hence called the DNA polymerase I or Kornberg's enzyme. There are other types of DNA polymerases such as polymerase II, 3, 4 and 5, out of which DNA polymerase I and 3 are involved mainly in DNA replication and the rest of the polymerases are involved in DNA repair. However, DNA polymerases requires a template DNA, short primers and a free hydroxyl 3' free end for polymerization of nucleotides during DNA replication. Let us discuss more about DNA polymerase I. DNA polymerase I enzyme provides the major part of activity in E. coli. It is mostly involved in DNA repair and is used for in vitro DNA replication. The main three activities of the enzymes are: number one, the enzyme has 5' to 3' polymerase activity, which is responsible for DNA synthesis or also known as primer extension. Secondly, it also has 5' to 3' exonuclease activity, which is involved in removal of newly synthesized DNA strands during DNA repair. The exonuclease activity removes 10 bases at a time. And in the third, the enzyme also has 3' to 5' exonuclease activity, which is responsible for removal of nucleotides during proofreading. During proofreading activity, only one nucleotide is removed at a time. The polymerase action during replication does commit error in DNA synthesis, which need to be corrected. DNA polymerase is known to analyze the new bases added to the growing chain and to delete or remove the wrong bases. This is called proofreading, which reduces the error of replication by more than 100 folds. DNA polymerase I is encoded by a gene Pol A, which codes for a single polypeptide. The enzyme can initiate replication in vitro at a nick in a DNA duplex in a process called nick translation. The process of nick translation helps in removal of RNA primer and DNA synthesis on the lagging strand, also known as the Okazaki fragments. If the 5' to 3' exonuclease activity is removed from the enzyme, then the larger fragment is called clenofragment. In DNA polymerase III, DNA polymerase III holoenzyme is the most important enzyme that is required for DNA replication in prokaryotes. It was discovered by Thomas Kornberg and Malcolm Gefter in 1970. Along with the polymerase activity, the enzyme has proofreading activity that contributes to its high fidelity in DNA replication. DNA polymerase has alpha, eta, and theta subunits as the main replication components. The alpha subunit is responsible for polymerase activity. Eta has a 3' to 5' exonuclease activity, whereas that theta has a stimulatory effect on the proofreading activity. Apart from it, the polymerase has two molecules of beta subunits that serves as clamps that keeps the polymerase bound to the DNA. It also has two tau units that dimerizes the core enzymes. Another subunit of the enzyme called the gamma unit serves as clamp loader for the lagging strand. DNA polymerase III synthesizes DNA strands on both the leading and the lagging strand. Due to its high processivity of around 1000 bases per second, the activity of this enzyme remains vital in DNA replication. The next enzyme that is very important in the prokaryotic replication is the Primus. DNA replication cannot be initiated without the presence of a primer, as DNA polymerase is unable to start its synthesis without a free 3' OH end. Primus, also known as DNA G enzyme activity, catalyzes the synthesis of RNA primers to initiate DNA replication. In eukaryotes, the activity of DNA polymerase alpha provides this function. DNA G protein synthesizes a short RNA primer at the origin of replication, which serves as the primer for leading strand synthesis. In the leading strand, a one-time synthesis of RNA primer is enough to replicate the leading strand, whereas short primers are synthesized by DNA G for every few kilobases, which functions as starting point of the Okazaki fragment in the lagging strand. DNA G interacts with DNA B helicase to carry out this reaction, and the primer is synthesized in the direction opposite to that in which the DNA B helicase is moving. In effect, the DNA B helicase moves along the strand that becomes the lagging strand in DNA synthesis. However, the first primer let down in the first DNA G-DNA B interaction serves to prime leading strand DNA synthesis in the opposite direction. Deoxyribonucleotides are added to this primer by a DNA polymerase III complex linked to the DNA B helicase tethered to the opposite DNA strand. The another enzyme that is important in the initiation of replication in prokaryotes is the helicase. The ORIC of E. coli is recognized by DNA A and DNA C protein, which leads the DNA B, also known as the helicase, to separate the two strands. Loading of the DNA B helicase is the key step in the replication initiation. As the replicative helicase, DNA B migrates along the single-stranded DNA in the 5' to 3' direction, unwinding the DNA as it travels. The DNA B helicases loaded onto the two DNA strands thus travel in opposite directions, creating two potential replication forks. All other proteins at the replication forks are linked directly or indirectly to DNA B. An important enzyme, also known as Gyrase or Topoisomerase, is an important factor in prokaryotic DNA replication. DNA gyrase is an essential bacterial enzyme that catalyzes the ATP-dependent negative supercoiling of double-stranded closed circular DNA. Gyrase belongs to a class of enzymes known as topoisomerases that are involved in the control of topological transition of DNA. There are two classes of topoisomerases that play important role in DNA replication. Topoisomerase I. It is the first topoisomerase discovered in E. coli and removes negative supercoils. It cuts a single strand of the double helix of DNA and unwinds it to generate a relaxed negative supercoil by its enzymatic activity. The topoisomerase I acts only on negative supercoils but not on positive supercoils without the requirement of any energy. This enzyme is essential to relax the negative supercoils generated by topoisomerase II. However, its role in the replication fork is limited, as the growing fork generates a positive supercoil which is managed by the topoisomerase II enzyme. Now let us discuss about a topoisomerase II enzyme. The first type 2 topoisomerase, also known as Topo2, to be described was isolated from E. coli and named as DNA Gyrase. Topo2 enzyme have the ability to cut both strands on a double-stranded DNA molecule, pass another portion of the duplex through the cut, and reseal the cut in a process that utilizes ATP. Depending on the DNA substrate, these maneuvers will have the effect of changing a positive supercoil into a negative supercoil, or of increasing the number of negative supercoils by a factor of 2. Another enzyme called the ligase. DNA ligases cells nicks in the phosphodiester backbone of DNA. In the process of replication, this enzyme is essential for the joining of Okazaki fragments during replication and for finalizing short patch DNA synthesis during DNA repair process. There are two classes of DNA ligases. The first type of ligase use NAD+ as a cofactor and only found in bacteria. The second uses ATP as a cofactor and found in eukaryotes, viruses, and bacteriophages. The process of ligation by DNA ligase. The reaction occurs in 3 stages in all DNA ligases. First, formation of a covalent enzyme AMP intermediate linked to a lysine side chain in the enzyme. In the second step, a transfer of the AMP nucleotide to the 5' phosphate of the nicked DNA strand. And in the third step, attack on the AMP DNA bond by the 3' OH of the nicked DNA, sealing the phosphate backbone and resealing AMP. The next enzyme that is important in the termination of replication is the TUS protein and topoisomerase 4. During termination of replication, there is requirement to block DNA synthesis in one of the replication fork in the bidirectional replication. The third sequence, which is around 20 base pair sequence, are arranged in the chromosome to create a trap that replication fork can enter but cannot leave. The third sequence functions as binding sites for the protein TUS, also known as terminus utilization substance. The TUS-TER complex can arrest a replication fork from only one site and prevent over-replication. Only one TUS-TER complex functions per replication cycle. So, when either replication fork encounters a functional TUS-TER complex, it halts, the other fork halts when it meets the first fork. The final few hundred base pairs of DNA between these large protein complex are then replicated by an as yet unknown mechanism, completing 2 topologically interlinked or catenated circular chromosomes. DNA circles linked in this way are known as catenans. Separation of the catenated circles in E. coli is done by topoisomerase 4, a type 2 topoisomerase. In conclusion, DNA replication is an essential biological process that is responsible for transmitting genetic information from one generation to another. The process requires a close and intricate coordination of enzymes. Moreover, the process should be controlled and made free of error so as to keep the genetic information intact. This process is achieved by DNA polymerase 1 and 3 with its proofreading activity. Other enzymes and proteins such as ligase, topoisomerases, and helicase keeps the accuracy of replication by joining nicks, relaxing supercoiling of the helix, and subsequent unwinding of the DNA duplex to facilitate movement of DNA polymerase during replication.